Is it on the bread? Yeah. Mullet's got it.
Marco again.
Yeah, and you know what? I think he forgot what he was doing halfway through.
Did you ask him?
No. I asked him that, but then I think he forgot, and he just went back to cutting it like straight back. He just forgot halfway, and I was like, and like he started getting about the curve. Technically. Okay, I'll see him a bit.
Yeah.
Actually.
So this is Jake, my son.
Hey, good to see you, Jake. How are you? Okay? Right, tell me everything.
Gavin's lab thread.
All you need to know is,
All you need to know.
you get the order.
The order is
Can I put my laptop out for a few notes?
Yeah.
I just nice. I know you shouldn't really read notes, but I mean, I'm a bit rusty. Oh, cool.
So what we'll do,
And Andrew's after me, presumably.
Andrew's after you, yes.
I can just about read it.
So that's it, and I'll introduce you. But I won't say anything about your company.
Okay.
I won't say anything about your
You don't know anything about the company?
I know nothing about it.
You've got a podium if you want it. Yes.
Oh, okay.
Oh, that's right, I'll start off.
Yeah, click it here.
Click it there. Yeah, I'll probably put my laptop out just because I've got a few words that I want to hit.
This is, the key thing is the 7 minutes.
7 minutes. Yeah.
So just be very
Just push me up the stage.
right. Yeah, makes sense. I'll just go.
Do you want me to sit in the front? Just copy up.
What about yourself?
You'll be 8 minutes. Thank you. Sorry. There's a few more to come in, but I'm going to make a start. Oh, is that right? Is that okay? Is that right? I don't think that's Good morning, everybody. Welcome to Pivotal Links 2026. Can you all hear me? At the back, can you hear me? You can hear us. Oh, that's better. I should put it closer to my mouth. Obviously, I'm not used to doing presentations. Welcome to Pivotal Links 26. I think this is our number 15 or 16 meeting. There are one or two people who've been here from number 1. I see Nick over there and Ravi. So it's been going on. We had 8 companies on the first meeting, and this time we've got 85. So delighted with that. I've worked out stats on that, even I can work it out. We've got 20% growth on last year, which is particularly good because of the way the market's been recently. I'm just delighted that you guys have made the effort to come here and take up this opportunity to network. So we've got people from various countries who've come over from all over the globe, and we're delighted with that. I think this year we've got people from Asia, people from USA, people from Europe, and that's great. Sometimes we have less from different territories. So everyone's come this year, which is great. I want to sort of say a sincere thank you to our sponsors here. So you'll see all of them. They all have a booth. So please go along when you have got some time spare, when you're not doing your umbrella meetings, to go and say hello to them all. But we appreciate all of their support. For the first time this year, we've brought along a sponsor called BioDirect. BioDirect is a company set up by Pivotal Scientific. We have a booth in the main hall, and we've recognised that many life science companies such as yourselves have got very, very highly experienced teams, but they have certain needs which they don't have in-house. So what we wanted to do is to let you guys know that we have a panel of consultants, there's about 40 or 50 globally, that you can use for ad hoc projects. 3 of those people have come in today. They're available for networking, and we have a booth over there which will tell you how to do it. So these are very, very experienced, vetted by Pivotal Scientific, and these people will be able to give you good service on an ad hoc basis. We have a couple of practical points today. I have meetings with some of you, and I've got my own pod. I'm lucky enough to have my own pod, which is out in the reception area. So if you go to the main hotel reception, there's 2 or 3 pods in a row. I'm in the first pod. So if you've got a meeting with me, that's where you'll go, into there. Everyone else's meetings are on their tables if they booked a table. If you haven't booked a table and you don't know where you're meeting, then you go to the Pivotal booth at the front, and that's where you'll reach your delegate. The meetings are, we've been here before, 20-minute meetings, which sounds, oh, that's not enough. It's normally enough to get your points across. And then if you want further conversations, there's plenty of informal opportunities to talk. We had a lot of informal opportunity last night. Thank you to Dan and ARP for sponsoring the wine event last night. That went down very well. Those of you who can remember it, it was a fabulous evening. And there'll be an informal dinner tonight. We've changed it slightly. So I don't think UK is known for its food, I would say. So we've decided to do, rather than pretend to cook Mediterranean food badly, we've done a British theme menu. Okay, so we've got a British themed menu, so hopefully it will be better than previous years. And so we decided it's going to be, rather than a gala banquet, it's a British menu, British theme. You can wear whatever you like. You don't have to dress up. If you want to go and get changed and wear jeans and things, absolutely fine. Whatever you want. It's very, very casual. And then, of course, tomorrow the networking continues. There'll be lunch area, which you go and have your lunch, obviously, which is upstairs in the main restaurant. That's a buffet-style lunch. You can continue your meetings there if you want to. And, of course, tea and coffee throughout the day. I just wanted to say one fact. So we've got 132 people attending today. So if each of you just find one valuable opportunity, that's 132 valuable opportunities. Hopefully you'll find more than one valuable opportunity today, but I just wish you all success in the day, and I hope you have a great meeting. Thank you very much. So, now I want to introduce you to our first presenter, which is Stefan Hoyle, and he's a senior consultant for Pivotal Scientific. Stefan.
Thank you, Tim. Good morning, everyone. Last year I presented on the state of the life science market, and I'm not a scientist. And it's because our industry isn't just about science. In fact, today I'm the only non-doctor presenting. So you will hear some big words that I use randomly. In no way do I suggest I understand what I'm referring to. Jake, which way is it? Thank you very much. Tim, you can start timing now. We are all timed. Predicting our industry is a little like predicting the weather. Despite having extensive, and there's my presentation. Despite having extensive knowledge of weather, just a few factors can greatly influence the outcome. PSL, when we think about strategy and we think about our industry, we look at patterns. We look across science, we look at the commercial and customer, and we aggregate all of that data to deliver our view of how our industry evolves. And today I'll give you a sense of how we're seeing our industry over the course of 2026. Now, of course, many of you will remember last year I spoke about trade policy, specifically USA. But actually I should refer to trade policy in China and the USA, because both markets have significant impact on our industry. We all know about the volatility. The most interesting dynamic is how we have adapted to that market. The biggest change, I think, is not that, but it's AI technology. It is our very strong opinion when you think of translational biology that we now see workflows that go from the bench to the bedside. And when I've raised this recently, from bench to the bedside, I get a lot of, no, we haven't, that's not happening. I would argue we have enough evidence today that the research we do today is more quickly being rendered at the bedside. And as I drove up, I was listening to Radio 5. It's a UK radio station. It covers news and sport. And in the 90 minutes, they covered 4 topics that involved AI. Robotic dogs at the Soccer World Cup. But they covered ADCs. They were discussing a new drug that they brought to market that improves chemotherapy. It reduces the cycle and it improves life expectancy by 30% for women with late-stage ovarian cancer. And the reference was, wow, we're talking about something that's here today. And that's what I mean by workflow. We're moving, we think, from a very silo-specific environment to a workflow that's highly connected, highly collaborative, where we combine AI, multi-omics, and very rich big data sets. But workflow is not just about science. What we've seen at PSL is an evolving supply chain, where the expectation is it's both digital and traceable. We've seen it in the wet lab, where it's no longer just about trial and error. And of course, we're seeing it in production with our most precious resource, people, where as a consequence, we are seeing people's role change from producing to where they are doing governance, oversight. Every year we reference sustainability, but what we're now seeing is very clear evidence of green labs or cell-free biomanufacturing. And of course, with regulation for those in IVD, this risk-based approach to rules has completely changed our industry. Even grandfathering of existing legacy products used in IVD now require compliance. Now, last year I presented a list of trends. What I didn't do is explain the relationship between these trends. And it's our view that what you're now seeing is that AI is no longer about novelty. Now, we know it's not perfect. I have this, which tells me how many steps I do. I have this that tells me whether my heart rate is working. I have this that tells me if I'm going to die in the next 5 minutes, 10 weeks, 10 years. We know it's not perfect, but what we're seeing is this evolution and role of AI. If you bring together biology and you bring in data sets, all of a sudden through multi-omics, through multiplex assays, you really start changing that bench to bed workflow. It links our basic research with clinical outcomes in a fraction of the time. My reference to the comment they made this morning on the ovarian cancer drug, the ability to bring it at a fraction of the time than it was just a few years ago. We think bench to bed is driving a change in our customer need. The need for raw materials, the need for advanced technologies to deliver a much more sustainable, high throughput clinical outcome. I sit on the board of a CDMO, I sit on the board of a bioprocessing business. Both of those spaces are seeing double-digit growth. Albert Einstein once said, research is what I am doing when I don't know what I am doing. And the argument is, I think research is evolving at a far faster rate than we imagine. Now, of course, revenue, we have seen revenue growth in Europe and the USA. In fact, I think most commentators would view revenue growth in Asia as being the fastest moving. But this year, we've started to see growth in R&D. I've already highlighted multiomics and data, and the point I wanted to refer to was the emergence of the specialist. Our hypothesis is domain experts, especially in new tech, are performing materially better than your traditional broad-based manufacturer service partners. And I've seen that in the examples I'm working in today. We're seeing in M&A increased work. We're seeing both buy side and sell side both now re-entering the market. There is a money inflow. But the one I want to refer to is at the bottom. If we could reuse, represent, or re-engineer our existing product, and that is what we're seeing, just think of the opportunities that presents us. Because the 1+1 principle, which we've seen in 50 to 60 transactions, says the majority of our customer base consists of one customer buying one product once a year. And what we're seeing is this is not just about new technology. It's about reusing our existing technology, changing our commercial model, and representing that product to our customer. So I go back to AI, the model that we know is not perfect. Philip in the front row here is a lawyer. We were talking about the impact of AI in law. And there are firms today who believe they no longer need to recruit individuals because they've got AI. And we can see here an example in biopharma of where AI is being deployed. But what I'm most interested in is the box enterprise and operations. Because real change starts to come when you change your own internal processes. Tim yesterday gave me a document in advance of a meeting we had that gave me a whole list of questions that he generated from AI to work out what we should ask. Now, we knew all the questions. We knew exactly what to say. We probably both concluded that it wasn't that we were asking the wrong question. Maybe we should be asking, what risks do we see? But in 2 minutes, we generated something that would take us 20 minutes today. If you then apply AI to RUO and IVD, we believe that customers now require new offerings from domain experts, especially when you think about translational biology. But actually the question I want to raise is at the bottom, and that is around the broad catalogue providers. Unless they tell a really clear story on the specific pain points their portfolios have, they will be left behind. Now, we already see great evidence of catalogue providers who are improving, who are representing, who are re-engineering their legacy product and taking that to market. Last year, I presented this slide, and this was a slide comparing it to my old industry. I spent 20 years in telecoms and IT, and it was comparing how we were performing. And this year, it is very clear we're performing far more effectively, both in our consumption, our manufacturing, consumption, R&D, when I compare it to my old industry. But the two messages I want you to consider is AI has yet to truly scale in the enterprise, not in discovery, not in the workflow, but in our own businesses, how we consume the technology to help us. I have a friend who's a director of a very, very large consultancy practice, one of the big three. They now get so many requests for AI that they now offer two offerings, an AI-only derived consultancy or AI and human. So we know we're on the curve where the combination of the two technologies really works. But it's the second thought I wanted to leave you with. Between 24 and 25, we had 5 to 6 transactions. So far this year, we've nearly doubled it. That is twice as many people wanting to talk about M&A than have done in the last 2 years, and we can see that trend upwards. So, our conclusion. We recognise research is evolving quickly, and it's increasingly bench to bedside. AI is changing our R&D, our internal workflows, and what customers demand. But it's not just about the new, it's about re-engineering what you've got and representing it to your customer. Thank you.
Thank you, Stefan. 10 minutes it took there on 7, but however, very good. Thank you, Stefan. Next in line will be Jonas Funk from Ultrabright, a Munich-based company. I can't really call you a startup, but you're part of a bigger organisation. But
perfect, thank you. Okay, so we are Ultrabright Biotech, and at Ultrabright what we're doing or making are ultra-bright antibodies for high sensitivity assays. So what you can see here in blue is an antibody, all of you know this, and the antibody has a couple of signal-generating labels attached, right? And these types of detection antibodies are core to many different assays, including immunoassays, flow cytometry, or imaging. And they're both used in the research field as well as in the diagnostic field. So, but the problem with these conventional detection antibodies is they only carry a limited amount of labels. Typically, you get like 5 to 7 if you have a good label yield. Now, the problem is that this generates not enough signal for many applications. So the signal often is limited, and this limits the sensitivity of your assay. So often, low one targets are missed. Early disease detection sometimes becomes very difficult, and sometimes also you need very long incubation times in order to generate a signal. So what we at Ultrabright developed was this Ultra Beacon here on the right. It's a DNA nanoparticle precisely engineered, and we take the same labels that you usually attach to the antibody, and we attach those to our DNA nanoparticle. The DNA nanoparticle, the Ultra Beacon, has exactly one antibody attached and preserves the biophysical properties of the antibody. So all the rates are the same, don't get aggregation and so on. So basically, in a nutshell, this allows you to improve the signal in your assay up to 100-fold. So it depends on how good your conventional antibody is, so we can get 100-fold more signal. So the nice thing is you don't need to buy new instruments, you don't need to change your assay. You just swap the label, use our Ultra Beacon, and you get 100-fold more signal and sensitivity. So I just want to walk you through a couple of examples here. So here's a standard immunoassay where we capture an analyte that is analyte in yellow, right? We capture on a capture antibody, and we have a detection antibody that is labeled, and the detection antibody generates the signal. So what we do, we take the same two antibodies, same capture antibody, same detection antibody, we label this with our Ultra Beacon. And we can tune the number of labels on our Ultra Beacon. We can control this very precisely. So if we say we have one label, we have one. If we have 200, we have exactly 200. So what we did is we took this assay, and what we plot here is the sensitivity of the assay. So that's really 1 over the lower limit of the detection, versus the number of dyes or labels on our Ultra Beacon. So if we only have one label, right, we are worse than the conventional. So the conventional in this case had like 7 labels. But as we increase the number of labels on our Ultra Beacon, the sensitivity of the assay increases. So in this case, we got 23-fold increase in sensitivity, and this is just by using a different detection reagent, changing a consumable. You can also use our Ultra Beacons in flow cytometry. So here's a staining of rare antigens on cell surfaces. This is a conventional one that you buy off the shelf from some of the big vendors. And you don't see a difference between the isotype control and the staining, right? But again, if we use the same clone antibody but conjugate our Ultra Beacon, we can really nicely see. Now you get a signal that actually get a shift in staining index, so you can really separate the two populations. So this would allow you to sort of maybe pick out very rare events or low expressed targets. And we also tested our Ultra Beacons in spatial biology, microscopy, or IHC. So here's a tonsil slide, then we stained with a primary antibody, and then streptavidin, and we did the same with the ultra beacon, but has streptavidin. And again, I think you can clearly see this is here on the right side. You can see some things, you can see the cells, and here you don't. So they are sort of what I've shown you, I guess, is that you can use these ultra beacons and get up to 100-fold increase in sensitivity and signal. They can be seamlessly integrated into existing assay platforms and workflows, no new devices, and you can transfer them from one assay to the next assay to the next assay. That said, we have a couple of sort of products and portfolio. So we have some off-the-shelf solutions that we offer to our customers. So we have a set of secondary binders with antivirals, anti-human, streptavidin. We're always trying to expand this portfolio to meet sort of the demands of our customers. And then the customers, depending on the application, right? If you do flow cytometry, they want fluorescent dyes. So we have a range of different fluorescent dyes that the customer can choose from. But we also have chemiluminescent dyes, so actinium, ruthenium based for immunoassays. And then we also have enzymes such as HRP for stabilizers. And then also we do a lot of custom projects with our customers. So the customer has a special antibody they ship to us, and we conjugate it to our ultra beacon and ship it back, and they use it integrated in the assay. So just a couple more words to Ultravite. Ultravite was funded last year. We're a very young company. Was funded by Benjamin, Klaus, and me. Benjamin is also here today, by the way, as a spin-out from Platonic Biotech, which is a therapeutic company. And yeah, we are all experts in DNA nanotechnologies. It's the core technology we use to assemble our ultra beacons. And then we are based in Munich, Germany, and have a big lab or access to a big lab and production infrastructure. And we are, since like 1 year that we started, we have growing customer base, both in research field, but also in diagnostic. And in pivotal links, we are happy to be here today. I mean, yesterday was pretty nice. And so what we are looking for basically is any type of partner relationships where we can develop new research kits or diagnostic applications, but also we're looking for suppliers for antibodies, dyes or labels, and also for investors for upcoming financing round. So happy to talk to. Originally, Klaus wanted to come here. He has some personal matter he needed to attend, so he booked a lot of meetings. So I'll be taking over the meetings, and also Benjamin is here. So we are happy to connect this meeting, and yeah, looking for a great meeting. Thank you.
Thank you, Jonas. 7 minutes, spot on. Well done. Very difficult to do 7 minutes, I must say. Next talk is going to be by Matt Wilkinson. There he is, up there from Striven. And I won't go into it about it, but we actually use the technology he's going to tell you about. We use it Pivotal very, very extensively. So over to you, Matt.
Thank you, Tim. And I just need a clicker as well. So everybody says they know their customer, and yet every engagement I've had over the last 7 years since I founded Striven, I've found that sales, marketing, and product can never agree on who the customer really is and what is most important to them. We started off as a life science marketing consultancy, and over the years we've worked across the intersection between science, marketing, and technology. And of course, the AI revolution has directly impacted everything we do, and so we jumped on quickly. And of course, I could be talking to you about AI discoverability or AI adoption in commercial teams. But what really excites me, and where I think one of the things that is massively overlooked at the moment, is how we can use AI to get closer to our customer. Every engagement I've had, I must have had hundreds, thousands of conversations over the last 7 years. And at every one of those, the projects start with the customer at the center. We go off and we get voice of customer. We use all the information that's in the CRM systems that's within the team. And over time, the customer gets reduced. It goes from being detailed research to being a persona document that gets put in a drawer and forgotten about, stored in a hard disk drive and rosts away, where it might actually end up just as a quote in a PowerPoint presentation. And I think that's a real miss, because we're all here to serve our customers, and our customers are doing life-changing work for so many of us around the world. And so we realized with AI that we could do something about it. But before I tell you what, I kind of want to give you a little insight into where the customer leaves the room. And so typically 3 places. The customer becomes a slide. Rather than a real person as we're making decisions, we reduce them and we reduce their insight. Marketing writes from the inside out. What do I mean by that? Well, anybody that's ever tried to write marketing copy knows how hard it is to put yourselves in a customer's shoes. The customer might be a size 5 shoe. I'm a size 12. That makes it really difficult for me to walk a mile in a shoe. Yes, I'm a recovering scientist. Yes, I've walked a little bit in the lab, but it's been a long time. It's been maybe 2 decades since I've really worn a lab coat in earnest. And so it's really hard for me to understand exactly what motivates them today. And then sales meets the customer cold. Yes, they might have the sales scripts from the sales enablement stuff, but they haven't necessarily embedded all of that knowledge into what they're doing. I gave a talk in Boston late last year, where I met Tim. And one of the really, really funny things was I created a whole scenario, and I created a role play based on a ton of research that I put together, and then I failed it because I hadn't learned it. So I then showed how you could use AI to learn, and then sort of actually use an AI to test how well you learned your sales scripts. But sales tends to meet the customer cold because they haven't got a representation of them, particularly when they have, when they're moving into new markets. And I don't think this is a person problem. I think it's a physics problem. It's something that I like to call organizational gravity. My dad reminded me the other day that I've been obsessed with gravity since the age of 4 when I asked how do rockets leave the planet if they've got gravity on them. Clearly I misunderstood things at the age of 4. But what we find is that in every interaction internally, whether it's a decision-making process, whether it is a review process, every decision seems to take the messaging, the decision away from the customer and bring it back into the organization. And so the question I've been asking is, can we do anything about this? And the answer is yes. So for the last year or 2, we've been building synthetic customers. Now these aren't just going into ChatGPT or Claude or Copilot and saying, hey, operate as a whatever. This is using your own voice of customer or the research that we can find out about people online, because we can do a really good job just from actually looking about what our customer types are saying online. And we can build these synthetic customers. Now this is a synthetic customer that I built for a client. She's Priya, and she's dialed in specially to talk to you all today. And it's been, because she's got an American accent, clearly she's got up in the middle of the night because she's so excited to speak to you all. Priya's built on a lot of qualitative research as well as some quantitative research and some research we did on the lab. And I gave her a scenario, told her that we'd done the research and she'd been involved in that qualitative research. And then I sort of gave her a pitch that the company, a brief that the company was going to use to create a marketing plan. And then I gave one from a competitor. And here's her response.
Researcher gave me 90 minutes in March. I told her about the workflow that keeps me awake, the support gap that lost us a method transfer last quarter. I thought maybe this time someone was actually listening. I saw the launch yesterday. It was a really good presentation. The problem? Sadly, none of it was for me. I was asked the same questions by your key competitor, the product they just launched, and all their messaging. It all felt as if it was designed for me. No prizes for guessing who I am going to buy from.
This isn't a criticism. This is actual evidence speaking back to you. This is your own voice of customer, your own insights that everybody in this, this room holds about their customers. And what we do is we bring that alive and we allow it to then react to what you're thinking about doing, whether that's in product development, whether that is in marketing or even in sales. We've been working with product teams, marketing teams, sales teams, and one of the most exciting things is seeing how this can absolutely change the way that the customer becomes part of that conversation. And let's be clear, I'm not advocating for getting away from voice of customer. I'm advocating that we need to do more of it. But what I am also saying is that if we're going to do more voice of customer, if we're going to get to know our customers better, we need to make more of that insight. And this is a way that we can really bring the customer alive at 10 o'clock at night when we're trying to make those last minute decisions about something where we can't go and actually test this with a customer live before the conference. So if you'd like to find out where your buyer is going missing, I'd like to offer you all an opportunity. I think I said 30 minutes, but apparently it's 20, so I misread the script there. So 20 minutes and 7 questions. And I'll be able to go through a short diagnostic and be able to help you identify where your customer's going missing. And hopefully come up with an idea of how you can keep the buyer in the room. Thank you very much.
Thank you, Nat. Very, very good. So all of these presenters you can speak to after, but we won't ask questions as we want to get this all over and done with. Next talk is going to be by Gavin Alsop from LabThread. There he is. Thanks, Gavin.
All right. Thank you. All right, 7 minutes. I'll log that as a run. It's been a while since my heart rate was pretty high anyway. So there we go. Okay, so. It's going.
Oh, sorry.
I don't think I am
gone
next.
the wrong
I'm
way.
first.
Sorry, my fault. It's Tim from Invivo. I'm Tim. I only have one job.
So Gavin, good job. So thank you, Tim, giving us the opportunity to present on stage here about Imbue Biotech. My name is Tim Melsing, I'm head of R&D. 7 minutes is tough, so let's go. Who are we? We are one of the largest contract and development manufacturers in Germany for bioproduction, nearly 30 years experience. We implemented the cutting-edge antibody discovery technology using the Beacon in-house. And further, we have several opportunities for pilot studies, upscaling for preclinical studies, and IVD raw materials. So we are located in Berlin, in Germany, quite north of the city, in the new Berlin Brucker campus over here. One of those buildings is our bioproduction site. And our main business is pilot and upscale manufacturing of proteins and antibodies, novel antibody discovery, and several catalog products. So in the pilot and upscale manufacturing, we focus on several areas. It's the monoclonal antibody production in the bioreactor scale, batch of perfusion processes. In the recombinant protein production field, we use transient systems or CHO systems. In E. coli, we can go from shake flask to bioreactor as well. We do stable CHO cell line development and the monoclonal cell line stage, pharma grade, and GMP ready. Further, we are providing immunoassay development, immunoassay manufacturing, and developing and manufacturing custom immunoassays. So let's head into the novel antibody discovery technology we implemented. This is a beacon device from Bruker. Some of you might have seen before, and it's really sophisticated. It's moving cells by light into those nanopens, so it's very, very tiny wells, up to 20,000 on one chip, which allows true single-cell functional screening assays. This is really important during an antibody discovery approach, that you can already screen the antigen reactivity on a single-cell level. So this is made for a single B-cell antibody discovery approach using, for example, rabbit B-cells, using mouse B-cells, using llama, or using even human B-cells. And that's quite important to use B-cells having access to native immune repertoires. There is no need of display technologies. And what I said before, it's really important to have this function-first screening opportunity in the beacon. We preserve natural heavy and light chain pairing, and you can also perform cell-based assays into these channels over here, not only by a peat-based assay. You can also include native cells having a surface antigen native on the expressed on the surface, so you can have a true function-first screening. This will rapid the timeline massively. You only have to export, let's say, the correct B cells and the right B cells to be sequenced and analyzed further. So this is an important topic as well. As I said before, you only need some hits in your campaign to have a really, really good antibody. So it's okay to have low abundant, low frequency antibodies in your B cell population, and this is really possible with this approach. So what's also possible, we can have a cell line development approach and T-cell profiling. If you want to know more about that, please reach out to us. What else do we offer? We offer catalog products. We presented the last year and the year before. Those are cancer biomarkers, and in vivo animal study portfolio, corresponding isotype controls, and neuroscience biomarkers and antibodies. We have very high quality standards, just capillary gel electrophorus, analytic cell measurements, analytic NCC, but also application testing in ELISA Western blotting, SPR, FACS, and immunofluorescence, where this is suitable. So collaboration on those catalog products is highly appreciated for characterizing using your method. So please reach out to us if you want to check those antibodies. Thank you very much for giving us the opportunity. And we are around. Please meet me or one of my colleagues.
Thank you, Tim. Sorry for getting told that side. So now I am going to get Gavin up. His turn, and Gavin is from a company called LabThread, a startup company. Thank you, Gavin.
Appreciate it. Right, 6 minutes 58. Thank you. Right, well, thank you for the organisers. Thank you for inviting me, and especially for giving me the best wine of my life the night before my presentation. I would say I'm more of a beer man, and I felt out of place, and it was really quite telling when I ate the butter thinking it was cheese. So thanks very much. So that's a little bit about me. So I just wanted to tell a story. This is a startup company. No one's going to have heard of it before. So I'll just tell a bit of a story, see what you think. If you like it, come and speak to me afterwards. But I actually met Tim Pivotal 2011. This was, so just after my PhD, I worked for a very small company called Pedrilla, and we made antibodies. So I would work in the lab, I would buy the peptides, purify them by HPLC, conjugate them, inject them into a mouse, to make an antibody. I did that. But everything I did was written in lab books. Everything was handwritten, printed out, stuck in the lab book. I used Excel files. It was good fun. Years later, the professor, the CEO of the company, would still call me and say, Gavin, where are these samples? How do I sort of find this product for my customer? Even though it was meticulously written in that book, he still called me. And I just have a question. If the founders and CEOs in here, how many people could rely on someone like me who would still take your call and help you out years after they leave the company? And if that doesn't exist, how much data and knowledge from your company might have been lost? Anyway, fast forward, Pedrilla was a long time. I got trained up by Thermo to be a CRISPR specialist, so I used to give seminars around Europe to give seminars on CRISPR. I haven't actually given a talk since 2018, so I'm a bit rusty, but you can see what I used to be doing. I found a guy called Ryan Kaywood. So he did his PhD in Oxford, and he started a company called Oxford Genetics, or Oxgene. He hired me because what he did at the company started out as just a plasmid company. He found a way to automate the approach of knocking genes out in cells using robotics. So he had 7 Hamilton-style robots. He had 50 staff, and I was selling that service for him. And he needed to find a way of capturing all the data, project managing up to 50 scientists, and handling all the samples. And he tried all the software that he could on the market. Nothing really stuck, nothing was scalable, nothing was easy to use. And so he actually employed a whole team of developers to build something bespoke for him. Anyway, fast forward, Oxgene was sold to WuXi, did pretty well out of it. I then went into the cell and gene therapy industry and worked for various viral vector manufacturers. Until now, I'm now a consultant, so I actually help companies, startup companies, develop their story, help build business development teams, and get their product to market. And he actually called me and asked me to do a little bit of research. He wanted me to go out and find if a product like this, an electronic lab notebook, a piece of software, was interesting and could be profitable. And I went into 10 biotech companies with my network and 5 universities and did some research. And what I found was sort of astonishing. So when I did my PhD, I spent months going through lab books of all the people that had been before me to find the origin story of plasmids and cell lines so that I could figure out whether they were plausible to use or not. That was 2010 when I finished. When I went into the universities, people were doing the same thing. And with the churn, with every 3 years, students are leaving and new postdocs are arriving, how much data and knowledge is lost? How much of the nuance of each experiment is lost each time? And I'm astonished that that still hasn't changed in academia. Whereas in biotech, it's more than implied you have to use an ELN and sample management tool. Yet all the companies I spoke to weren't so happy with either scalability of the platform, the usability, or there was something else. And that really got me thinking about what they're developing here at LabThread. And actually why I decided to join is their, well, I'm actually Chief Commercial Officer now, which is pretty cool, but it's only in Thailand. So anyway, let's talk about LabThread. So what I really want to say here is you've heard the story, you've heard of sort of the data that I found. And really, this platform is what I wish I had back at Pedrilla. And a lot of the companies I spoke to have tested this and really like what it is because of its simplicity. So it's an integrated electronic lab notebook system. It's a project management system. It's a sample management system and a communication system all rolled into one. It's less, we're going to charge less than the price of Snapgene, less the price than most of the ELNs in the market. And the reason being is we already had a legacy product that we built upon after Oxgene. And so the product's all ready to go. And we, launched last month. We've got 10 beta users. And so far, I've been pretty pleased with what people are saying about it. So that's what it's all about. It's all about enhancing collaborations. So far, so good. Okay, so really, we've built upon these core principles. Simplicity, it has to be easy to use, and it has to be much easier to use than pen and paper and what's available on the market. Compliance is essential. It has to be, it has to work with all the compliances that you need for to living your IP or everything that university requires, such as the FAIR principles now. Continuity. This is all about capturing data in the lab and allowing nothing to escape from when one scientist comes and leaves. And finally, visibility. This is built so that the founder of a company can see everything and the scientist can see everything, and you can collaborate together. So let's have a quick look at the software. I've got some time remaining. So it's based on the principle of projects. The projects are not tying the strands of the workflow together. From this point, you can control everything about your experiment, put all your reagents in, and it's the starting point for looking around everything that you're going to be doing in the lab with all the reagents that you have. This is the molecular biology software. It does everything Snapgene can do, so there's nothing particularly surprising about it. And if you had a play, it would be very familiar to you. This is the ELN software. So you get a front page where you have every tool that you would expect from Excel, PowerPoint, and Microsoft Word, and it's actually got sort of Microsoft integrated within it. So you can control everything from here. You've got all these tools, such as actually having your 96-well plates, grids, Excel files. You can import, export. Everything is completely free to control, and you can always download your data in a PowerPoint presentation or in a PDF as well. And everything is digitally time-stamped. So if you want to use this to protect your IP, show when you've done a piece of work, or create a report for your clients, everything is time-stamped, so it's fully audit trail. The LIMS software is exactly as you would expect. You can use this to log every single sample in any type of tube you want. Everything is automatically digitally barcoded, and then we can now map freezer spaces. So if you have particular freezers in your lab, you can have a map of it, exactly where your sample is, which box it's in, and then you can put that back into your ELN system. Finished. So I think tying together is really quite a robust piece of software. But the critical feature is, rather than having to look through every lab book, every piece of every file, you can use the AI system to basically search for every experiment, every sample, every result, and it'll come up for you. Should make things pretty easy. So anyone can try it for a month for free. We've already partnered with IDT, so anyone who buys us will get discounts from IDT, and we're looking for any new partnerships today. That was very great. Thank you very much.
Thank you. Very good indeed. Right, our next talk is going to be Andrew from the company SciTab. Here he is. And Andrew, I'm going to pass over to you.
Thanks very much, Tim. It's really great to be back at the Pivotal meeting. So what I'm going to be talking about today is how at SciTab we help so research reagent manufacturers, vendors, distributors drive their success and innovation by using our data products. So at SciTab, really what we're sort of very much focused on, we're a scientific data company. We're really laser focused on extracting from the scientific literature the research products that biologists are using, taking that data and from this unstructured form into this structured form, and getting out not just what products are used, so what research antibodies, kits, instruments, etc., but also how they're used and providing and harnessing that data. We do it on across the kind of research reagent space, so everything from research antibodies, biochemicals, experimental models like cell lines, bacteria, viruses, and most recently into instruments. This is at a massive scale, so we can search for sort of over 18 million products at the moment, get at 40 million scientific publications, and we get at these via both the open access but also by partnership agreements with publishers to make sure we have the right access agreements. We get to preprints, and then we're also going to get to protocols. We found over 12 million citations, so each citation is where an individual catalogue research product is being used in a publication, and we've got to the level of the published image. So this is the scientific image in the paper down to the exact panel where that product was used. And we use that data to power our search platform, so we've just rebranded it as CiteR Explore. This allows scientists across the world to rapidly search this database of research reagent use and allow them to find and then evaluate the research products of their experiment, with the whole point of them avoiding buying the wrong product that's going to derail their experiment and cost them time. We're really proud. We support about 90,000 users a month from across all of the main universities. 4 of the last 10 Nobel Prize winners in physiology and medicine, their labs we had users for, and right up to the sort of largest biotech companies. And that's just a snapshot of the search platform. You can search the reagents, go to the product pages, look at the images. You can search the images, find the products that we use, and go to the product pages that way. And again, there's the free trial option, so anybody can give it a go. We heard about AI, the AI wave. One of the things we've just launched is an MCP AI connector. So for companies that have internal AI agents, they can connect direct to our database. And of course, that's where, automated labs, agentic AI, that's where there's going to be a lot of interesting growth, I think. So this is aimed at researchers, but right from when we founded CiteR more than 10 years ago, we've worked really closely with the reagent suppliers, many people in this room, and we formed partnerships, let's say some of which are more than 10 years old. And we work with both the really well-known, very large organizations, but also the real specialists that provide really important products. And we work with them in a number of different ways. So I'm going to highlight sort of 4 products that we do, give you an overview of those 4, and then just give you a little bit of extra information on 2 of them, that sort of new things that we're launching or expanding for Pivotal. The first thing we do is we offer what we call market data around reagent use in the market. If you're making investment decisions about right down to where to put entirely new, which countries to invest in, but also importantly, which products are going to have the highest impact for your organization, then, for example, 4 of the top 5 antibody companies use our data to inform their pipelines. So that's the market data, it's quantitative trend-based data. The second way we work with reagent companies, reagent vendors, is with our search engine, we can partner to make sure that we have the correct listing and maximize exposure of your products on our site. Something new that we're just trialing, inline advertising, which I'll just give you a little bit more information on in the next slide. And then finally, the 4th product is citation provision. So you've decided what product to make, you've sold that product, scientists have used it, they've successfully carried out great experiments, and then of course they publish that data. People coming to your website, or researchers coming to the website, potential customers are going to of course want to see that that product has been used in the literature. And for any company collecting and standardizing that data, especially when you've got to get both the citation and now it's really accepted that you need the published images as well, is a real time sink. And we have this, of course, entirely automated. So we license our data for vendors to use on their website, both to ingest it directly and put it in their systems or our website plugin. And I'll show you in a couple of slides a brand new version of that plugin. So these are our, say, 4 products. Just to give you 2 slides on a little bit extra. So this is the inline advertising I mentioned as a trial. The aim for this is to allow companies to promote products which might be new, might be really exciting for researchers, but haven't got any citations yet. So it offers companies a chance to show an inline advert with sort of title, text, image, and link. And so this is brand new, it's a trial, we're going to see how it goes. So if you're interested, come and talk to Jonathan or I. Jonathan's also here. Can't see where he is at the moment, but give me an idea at the back there. So that's the inline advertising. Then the other product that I mentioned, I want to give you a little bit more information on, again, just because we're doing something quite new that we're wanting to show off really, is the citation provision. I mentioned we can license that citation information images directly sort of downloaded, stored in the company's databases, but many companies don't want to maintain the database and the web tools to display and filter and search those citations. So we offer a web plugin, call it our citation widget. It's a simple line of code that's copied onto the product pages and then can access our databases in real time and show all that information to researchers. And we're currently in the process of redesigning it with the aim of doing sort of upgrading it in a few ways, providing more summary information, again, sort of both important for the researcher, but also for SEO. As I mentioned, published images, I think they're now so important for researchers making decisions. They really want to see how the product actually performs. And this is just an example of one of our cropped images. So you can see it's panel B of that particular figure from that particular paper used for that product. So we're making them way more prominent, providing summary information about how the product was used, the sort of scientific data around the application, the species, the dilution, and also where it's used. Researchers, depending on the geography, seeing that other researchers within their country have used the product can be a really important driver for sales. So providing more information on that, highlighting high impact upplications, new citations. And as I say, we've got key clients in this room. All of our existing customers, we're going to offer this, the upgrade for them to use within their existing package. And we've just literally started reaching out to them. It's the first time we presented these images outside of our current customers. And of course, we'd love some new people to trial it. The official launch will be in September. So yeah, we're looking for new clients that might want to give it a go. So just to quickly summarize, as I say, we're really focused on extracting this data. It's incredibly valuable, but it's really fragmented unless you collect it and structure it around where these research products are being used. We use that for our search engine and also now this AI plugin and for researchers, and then for the manufacturers, vendors, distributors, we provide the market data, our search engine listing, this inline advertising trial, and then our citation provision, which includes the web plugin that I showed the new designs for. We love the long-term partnerships we've built. We'd love to expand those. If you're interested in expanding the way we work with you, then come and chat to Jonathan or I. And of course, we'd love to form new companies who we've never spoken to. We'd love to speak to you as well. If you'd like more information, you can scan the QR code and download a document with more information on what I've told you, or probably the best way, come and talk to Jonathan and I during the networking. Thank you very much.
Thanks very much, Andrew. Now I'd like to introduce Hon Lung from YEX. He's flown all the way from Canada this morning, so I hope he's going to stay awake for us. Hon, over to you. Thanks.
Good morning, everyone from Toronto. So I'm a professor. I started my lab at Mayo Clinic, and there we would take surgical specimens for cancer patients, and we would implant them in the chick embryo to do immunotherapy testing. When I moved my lab back to Canada, I'm Canadian, COVID happened, and so began to answer that question that I've always had for a while is, what is it in the yolk that sustains the embryo and hence these tumors? And so during COVID, we discovered that there's a fraction within the egg yolk that actually does everything. And so fast forward to a couple of years later, I'm also the CEO of this company called YEX. We're named after the product. It's an acronym called, it stands for yolk extract. So, okay. So YEX is short for yolk extract. We are competing with fetal bovine serum and serum free media, but let me tell you about fetal bovine serum if you already don't know what it is. It's essentially cardiac puncture of a fetus, bovine fetus, and then you clot that blood, and you use that serum to grow cells, and you add maybe 10%, 5% to your liquid media, and that really is a very effective way, a growth medium, nutrient broth for growing adherent cells and mammalian cells and whatnot. But there are a lot of drawbacks with it, right? There's a lot of variability, a lot of hidden costs to using it. You have to test it, and then every time you deplete your FPS if you want to make antibodies, You have to test it again to make sure that's truly free of the bovine IgG contaminant proteins. So we've manufactured a fully GMP compliant drop-in replacement for fetal bovine serum. It's called YEX. And this is what it looks like. It's this beautiful honey-like appearance. It's gorgeous. And we sell in 2 different formats. Half-liter bottle, just like fetal bovine serum, but also we also sell 50 ml tubes of it. And our customers like it because they don't have to buy the bottle, alcohol it, freeze it down. It already comes like that. It's value priced, it's very competitive. Like I said, we're GP compliant. We make everything, we polish everything, finish the product in ISO 7 clean rooms, and we provide a full certificate analysis. So we're here today because our beachhead market is really to help companies reduce their costs, streamline processes for manufacturing, whether it be antibodies, recombinant proteins, peptides, viral vaccines, you name it. So this is some data that we've been getting from companies where costs really matter, whether they're making antibodies from hybridoma cells or CHO cells or HEK cells. You know, they all face these challenges, these hidden costs. So up the top here is one graph where you can see one hybridoma cell line, it's a mouse hybridoma cell line, and over the course of 7 days you can see when grown in standard media, which is 10% FPS, that's how well the cells grow. But when grown in our culture media, it grows the same way. And you know, the proof is on the pudding here. The yield is about the same in terms of antibody production. Below is a set of data they also looked at with a thinly curved cell line. So this is a hamster cell line, hamster hybridoma cell line. And they found that actually YEX provided a better growth advantage, which is crucial for them because you know, this is a slow growing cell line, doesn't always produce a lot of the antibody, but with YEX it did. And there again, you can see about 20 to 30% more high yield in the antibody maker. And these are all catalog products. So I just wanted to show you some testimonials from companies that tested it, right? We don't want to show you stuff from labs. So BioXcel, Wellburns tested it, said really nice things about encountered no issues with cell growth, productivity met our expectations, final purified product was indistinguishable from the controls. Controls meaning that antibody grown in their standard media. Middle, very interesting, the poultry livestock. They want to move away from embryonic eggs to make vaccines, and they want to move into cell-based bioreactors for making these viral vaccines, these poultry vaccines. Saying that when they looked at it to grow poultry vaccines in 3 different products, with avian reovirus to egg drop syndrome, the viral titers were the same. Whether it was the cells that were used to make the virus, propagate the virus, were growing standard media or a YEX, same viral titers, which is great. This has just happened last month. AstraZeneca, you know, they're facing challenges with transient or even consistent permanent expression of their recombinant proteins, such as bispecifics, or even just antibodies. And they said, you know, they see about 60% more production when they use YEX compared to standard media, fetal bovine serum. This is really big for them because they're really struggling with not getting enough production. So this is really exciting. And because of this partnership, we've started this new distribution agreement with the United States with Avantor VWR. So this has been really exciting. Why? So in the case of AstraZeneca, they when they make their antibody or recombinant protein CHO cells or HEK cells, there's a proportion that the antibody always ends up aggregated. And so there's that hidden cost of getting rid of that aggregated antibody, right? So with YEX, you don't have that issue, just like fetal bovine serum. Another important thing for them is they need to grow all their CHO cells in suspension. They don't want to grow any of their cells in adherent. With YEX, we have a formulation that enables nearly all the cells to grow in suspension, which is fantastic. We know that a lot of companies are using HEK cells, suspension HEK cells, to grow their cells. And the media can be expensive. You can use YEX also to grow those same suspension HEK cells. You know, we obviously tested all these workhorse cell lines, whether it be MDCK cells for making H1N1 or H5N1 vaccine viruses, HEK293s, Vero6, but also some standard prostate cancer cell lines, even ones that my lab uses, right? The growth rates are similar. We won't say that they're better, but they're similar. And this is another thing that's happened this last month. ATCC, they want to evangelize Yaks because they don't want they don't want academic lab to continue using fetal bovine with all its drawbacks. They really see Yaks as being that next generation replacement. And one of the things that the insights that they gathered is it's not just about cell count, it's about health. It's about what are the cells consuming? What's the nutrient source? And so I think, some of the SMEs are really looking at Yaks to kind of see across the whole product range within ATCC to see, what is good, which ones will thrive or be the healthiest when grown with Yaks. So I think my 7 minutes are up. But yeah, we're a young startup company based in Toronto. We're eager to work with companies that are struggling with some of the hidden costs with manufacturing their products. And another tidbit, we have people that are doing iPSC, I'm not a neuroscientist, iPSC-derived retinal and cerebral brain organoids. And for them, the big cost, this is literally a cost, is the cost of the synthetic media. We've been hearing anything from a half a liter bottle of $1,000 to $3,000, where you have to culture these brain organoids. You have to change the media every day, every 2 days at least. They're showing that you can use synthetic, which is kind of at a fraction of the cost to do the same brain organoid work. So anyways, thank you so much. Hello from Canada, and I hope to talk to any one of you guys later today.
Very interesting. The last but not least is going to be a talk by Atul Tandon from Neo Biotechnologies. Where is he? Here he comes. You might also know him as Madman.
Thank you. Thank you, Dave. Good to be here and good to talk to the people who are really antibody experts. And today is the time when I will be talking about the history of monoclonal antibodies. Monoclonal antibody technology was invented close to 50 years ago with a promise that the technology will produce a single antibody against a single epitope in unlimited quantity with lot-to-lot consistency. What we have learned in the last 50 years, the technology is absolutely perfect. Millions of antibodies, millions of monoclonal antibodies have been produced, and they are all single antibodies, single epitope, lot-to-lot consistency, no problem. Today, the market has millions of monoclonal antibodies existing, but there is one problem. Majority of the antibodies, and majority, my friends, being conservative, I'm saying more than 70% of the antibodies on the market today are bad antibodies. They are really worth not using at all. And this 70% is a very conservative number I'm giving you. If you talk to the people who really evaluate antibodies, and my friend is sitting here, Dr. Guido Sauter. He is the man who has evaluated thousands of antibodies on the market on thousands of human tissues, and he has rejected more than 90% of the antibodies. So the truth of the matter is, after 50 years, what we are learning, that the technology is perfect, but we as the antibody developers are doing a lousy job. We have done a lousy job picking the right antibody. It is all, when you are developing an antibody, it is all in what you end up picking. The mistake we all have been making all the time for the last 50 years, we were just looking for the final use of the antibody. I want an antibody for Western blotting, so I am just screening, looking for antibodies, Western blotting, flow cytometry, immunohistochemistry. We never cared. We never bothered. Is that the right antibody? Is that a specific antibody? When I say an antibody is a bad antibody, what it really means is that in addition to reacting its intended target, it is also reacting with something else. And that something else, the seller of the antibody does not know, the user does not know. Nobody knows that. And then after years and years of using that antibody, publishing all the papers, making all the diagnostic kits, then you realize, oh, this is not the right antibody. So my friends, it is true that thousands of antibodies on the market, but vast majority of them are bad antibodies. Garbage antibodies, and I don't need them in my lab at all. So, about 8 years ago, antibody manufacturers, they got together and they realized that there is a problem. Before we worry about the application of the antibody, we got to make sure it is the right antibody. And that group proposed 5 different methods. We call them 5 pillars for antibody validation. One is a genetic method, knockdown cell lines. The other one is an orthogonal method, very popular today, where you generate your antibody-based data, and then you compare your data with a non-antibody data. And the 2 data should match. I will come both to that, but these 2 methods, genetic and orthogonal, are very popular. And I will show you how a lot of antibodies on the market, they fail. If you talk to my friend Guido says, hey, these are trash. I don't want them to enter my lab at all. 90% of the antibodies, he does not have. And that is a true statement. So in addition to the 5 methods, here the genetic method, the orthogonal method, independent antibody, I'm not going into the details of all that. I presume you guys already know. But our company, in collaboration with CDI, have been working on a protein array technology for antibody validation. We have a microscopic slide, just 7.5 centimeter by 2.5 centimeter. And on this slide, we have more than 21,000 full-length human proteins printed here. And these proteins are printed in duplicate. So what you are seeing is more than 42,000 spots. And if you ever want to see this slide, I have with me, you can come and check with me. So, and this is covering, the 21,000 proteins here are covering 85% of the human proteome. So literally, human body, the entire human body is sitting on this slide. And this is the lemon test. Put your antibody here and find out what proteins it is reacting with. And I challenge you, bring any antibody on the market, put it on this chip, antibody will show you the problem. So this is the method we are currently using, protein arrays with more than 21,000 full-length proteins. And I will show you a good example. I have lots and lots and lots of examples I can show you. But let me show you a pan-IgG antibody, anti-human pan-IgG. Pan-IgG means the antibody is supposed to react with IgG1, IgG2, IgG3, IgG4. If you pick a commercial antibody and you do a typical immunohistochemistry, B cells should be stained. Perfect antibody, perfect. You can show it to any pathologist of the world. And just by looking at this, it's a very nice antibody, very nice. When you put it on the protein array, my friend, this antibody, in addition to IgG1, 2, 3, 4, it is also reacting with IgM. And nobody can catch it. A pathologist cannot catch it. And this is how And this is a commercial antibody today. The antibody that we have developed at Neo Biotechnology, you can see right here. Antibody behaves just like this commercial antibody, but it is reacting only with IgG 1, 2, 3, 4, no reaction with IgM. So this is the right kind of antibody. And the market is flooded, I tell you, my friends. I have lots of examples and I do not want, and I never take the name of the suppliers on the market. But the market has lots of antibodies like that. Another thing we have learned after 50 years, that rabbit antibodies are far better than mouse antibodies. And especially the recombinant antibody. Recombinant rabbit monoclonal antibodies are the antibodies of choice today. All the reasons are listed here for the brevity of time. I will not go through them, but the bottom line is recombinant rabbit antibodies are preferred over the mouse antibodies. Mouse hybridoma antibodies, instability, extra chains, they can die. Let me show you some of the examples of the antibodies developed by Neo Biotechnologies. And I'm giving you 3 examples: cadherin 16, colipase, and nepsin A. We are looking at different normal human tissues. We already know where the mRNA for these genes is existing. So cadherin 16 is primarily in the kidney, mRNA. We made an antibody, we selected an antibody after making thousands of them. And 99% of the antibodies we threw in the garbage. We picked the antibody, that antibody on a Western blot. We did the Western blotting on all of these tissues. The antibody is reacting only with kidney. This is what it should be. And then we also did immunohistochemistry on all these tissues with the same antibody, and the antibody is reacting only with the kidney, nothing else. We put this antibody on the protein array, the 21,000 proteins, it is reacting only with cadherin 16. So this is a kind of antibody we need today. The same example here is the colipase. Colipase is a gene, the mRNA is only in pancreas. When we did the immunohistochemistry, we did the Western blotting on all the tissue lysates, antibody is reacting only with the pancreas. We did immunohistochemistry, reacting only with pancreas. We did the protein array, reacting only with the colipase. So this is what we need. Another example is a nepsin A. Nepsin A is a gene which is expressed in kidney and lung, 2 places. We did the Western blotting, we see reactions only in kidney and lung. We did immunohistochemistry, same thing, kidney and lung. And when we did the protein array, it recognizes only nepsin A, nothing else. We have gone one step further. Now we are able to do the epitope mapping. Say for example here is a cadherin 16 full-length protein. This is what was used for as an immunogen for making the antibody. Then we use this antibody on a phage display library where we have more than 700,000 peptides precipitated with this antibody, and then we sequenced the peptides precipitated and did the epitope mapping. Now we know our antibody is reacting between amino acid 699 to 730. So thousands of our antibodies are now going through epitope mapping. And we will be showing you and sharing this data with everybody where the epitope of our antibody is. Market is hiding today. Market is hiding today even the immunogen information. We will tell you the final epitope recognized by the antibody. Oh, a lot of companies, including my companies, we say, it's a proprietary information, we cannot tell you about the immunogen. We will tell you the final epitope. So this is how the cadherin 16, this is, we know, the 15 amino acids our antibody is reacting with. Come and talk to me and I will tell you more about the meaning of the dark green and the light green. Dark green means antibody is predominantly reacting with these amino acids. Now, using these monospecific antibodies, we have been developing ELISA pairs. So these are all antibodies, the capture antibody, the detector antibody, both are monospecific, and we are finding the pairs, and such pairs we are calling them mono-LISA. Monospecific antibodies for ELISA. And you can see these are some of the very popular clinical targets: anti-Mullerian hormone, CA19-9, CEACAM5. These are all very important targets, and we have absolutely monospecific antibody pairs for these targets. Lastly, I can tell you that the market is very rapidly moving into multiplexing, and the multiplexing means that multiple antibodies are used on the same sample, whether the sample is a paraffin section or it is the single cells for flow cytometry or the ELISA. Multiple antibodies are being used, and if one antibody out of the multiple antibodies you are using is a bad one, your whole result is a bad result. One rotten apple will spoil the whole case. So what I can tell you today is that market is big, opportunity is very big. The multiplexing market, which is close to 2 billion, will turn into 4 billion by 2030. People are looking for good antibodies, and antibodies are not there, my friends. So the bottom line take-home message is that we need fully validated recombinant rapid monoclonal antibodies. And the other message is no single method, no single antibody validation method is enough. We need more than one method. I showed you the orthogonal method for protein, the PAN IgG, not enough. You got to do something more. Thank you very much.
Thank you very much. He wins the prize for the longest presentation as well. We knew that. Thank you, everybody. We've got 5 minutes now until your networking starts. You should all know where you go. So I hope you have a great day or 2 days. Thank you very much.
Well.